So hello all. Today let's see something interesting. This is quite a recent and an interesting topic to discuss. This is about CRISPR Cas9. So what do you mean by CRISPR? The term CRISPR? So CRISPR stands for clustered regularly interspaced short palindromic repeats. And Cas9 is an endonuclease. Now let's move on to the topic. So, do you find it weird that I've included all these images, irrelevant images, on my presentation? Yeah, I have included a couple of barcodes here. Any idea why? Okay, and the next image is even more interesting. Why would I add a picture of, a lineup of all the accused that are required by the FBI? Any idea? So this is something you come across in all, prisons. There will be a notice board where, they attach all the images of the accused that they wanted to, that they're looking for, right? So any idea why these images are included? Let's relate these images to our topic somehow. Okay, so fine, I do not want to go on to CRISPR and everything right away. So now let's have an interesting discussion. This is going to be more of a story, so pay attention to it. So this story begins somewhere in 1980s, okay, in the late 1980s, to be precise, 1987. So in 1987 there was this Japanese scientist, okay, so this Japanese scientist called Ishino. So this Ishino, he was working on E. coli. Okay, so he was studying the DNA sequences in E. coli, and when he was studying these DNA sequences, he came across something more, more interesting. So what he found was that in the DNA sequences, there was a particular segment where there was this particular interesting finding. So he found there were certain, repeat sequences. There were clusters of repeat sequences, and these repeat sequences, they were intervened by certain um, random sequences. Okay, so these random sequences, he didn't know what to call them, so he named them as spaces. Okay, so he named these as spacers. So this spacer was different from this spacer, and so on. There were a lineup of spaces. In between these spaces, he had what is called these short palindromic repeats. So there were these clusters of short palindromic repeats, and in between he found certain random sequences. Fine. So he didn't know what its purpose was, so he just left it there. And later on, similar finding was reported by a Spanish scientist. Okay, so this Spanish scientist, his name is Mojico. So he also came across similar patterns of sequences in DNA of a certain bacteria. Fine. And later on, all these, this Mojiko, he didn't stop it there. He also put forward a hypothesis saying maybe this is in one way, somehow it is related to the development of immunity in bacteria. So he just hypothesized that this could be conferring immunity in bacteria, but he didn't prove his claims. Okay, it was just a hypothesis that he put forward to. There were no proof or evidence to to substantiate whatever he said. So later on, there was this interesting incident that happened. So I would say this is basically the birth of CRISPR Cas9 technology. So this happened in French. Okay, so in France, a French yogurt company. Okay, a French yogurt company called Danisco. So they had a this was a very flourished yogurt company, very well known. But in the later part of the the later days, quite lately, they suffered a huge loss. Okay, so they were not able to identify the cause for the huge loss that they suffered, and later on they kind of somehow were able to pinpoint the cause of the loss that they incurred, and the cause was found to be you guys know that for yogurt we need certain bacteria to ferment it, right? So if something has to be, if yogurt has to form, there should be some bacteria that brings about the fermentation, right? So this particular company that employed a specific strain of bacteria, and that particular strain of bacteria, it didn't survive. So most of it, they succumbed to a viral infection. Okay, so basically this was a bacteria. Let me draw a bacteria. So this was a bacteria, and this was infected by a phage. Okay, so this was infected. Let me try to draw a phage. So sorry for my pathetic diagram. Okay, so phage looks something like this, and it will also have its own nuclear material. Okay, so basically this bacteria, okay, imagine this to be a bacteria. So this bacteria was infected by the phage, and this most of the bacteria, they succumbed. Okay, there were few bacteria which survived. Okay, and they were like, they didn't understand why this particular thing started happening all of a sudden, so they wanted to investigate. So they wanted to find a remedy to this, and they employed a team of scientists, okay, to work on this particular issue. So this team of scientists was led by someone called Philip. Okay, so this Philip, what he did was that he sequenced all these bacteria which survived. Okay, and he also didn't stop with that. He also sequenced these bacteriophages. So he was very inquisitive. He wanted to know what particular what is it thing made certain bacteria escape this viral attack. Okay, so he wanted to study extensively the DNA sequences in these bacteria and these phages. So what happened was that he happened to find that this particular bacterial sequences were also similar to the ones which were previously described by Ishino and Mushiko. Okay, so there were these intervening repeat sequences, and in between these repeat sequences, um, to his surprise, he came across certain spacers like the ones which the previous scientist claimed. He also came to found that they were certain spaces, but to his surprise, these one of the spacers, okay, let me use black here. So one of the spacer nucleotides, the sequences in this particular spaces was similar to that of the phage DNA. Okay, the phage DNA, that's why I've drawn that also in green and this also in green. So the phage DNA sequences and one particular spacer sequences, they were similar. Okay, so then he understood that probably this is how this bacteria survives. Okay, this is one of the, what to say, strategy that the bacteria employs to survive this phage attack. So now let's see how this bacteria, this particular mechanism of how this bacteria has developed immunity against this phage. Okay, so what this Philip studied, let's see about that. So what happens is when this phage attacks the bacteria, this phage DNA gets incorporated. Okay, so let me draw the phage DNA. The phage DNA, it is double stranded. Okay, but I'm just drawing it this way. So this phage DNA is double stranded, and that enters into the bacteria. Okay, so what happens is this phage DNA will have certain sequences like this. Okay, let me draw. Yeah, so it will have certain sequences here. Okay, or wait. Okay, so this is one sequence followed by, yeah, this sequence here. Okay, so this is how the phage sequences are. Okay, and this particular what to say, sequence of the phage DNA, it gets cleaved, and a part of the fragment of this particular phage DNA gets incorporated within the bacterial DNA. Okay, a part of phage DNA gets incorporated into bacterial DNA. Okay, let me give you a real life scenario. So imagine this is I hope many of you would have seen this famous movie called Harry Potter. So I am a Potterhead, so I would like to relate this to the movie Harry Potter. So imagine, so who is the protagonist of Harry Potter? It's of course, as the name suggests, it's Harry Potter. So imagine this particular bacteria is Harry. Okay, and this phage here, this is the villain, right? This is the antagonist. So who is the antagonist there? Voldemort. Okay, so we have Lord Voldemort. So Voldemort is trying to kill Harry, and and Harry is trying to defend himself. Okay, so now let's see how this defense happens. How Harry is trying to defend Lord Voldemort. So what happens is when what do I say? This phage attacks a bacteria. A part of the DNA gets inserted into the bacteria, and the phage DNA is broken into small fragments, and one particular fragment is incorporated within the bacteria. So far, so good. So let's move on. What happens? So this purple thing here, okay, so this purple sequence here, this is called as, so this we already said this is spacer. So once it gets introduced or incorporated, so this is called as a spacer, right? So this is a spacer, and this is a short palindromic repeat. Okay, and let me call it as CRISPR only. So this is a cluster of short palindromic repeat sequence, so I'll call it CRISPR. And this is another spacer, and this is another CRISPR sequence. Okay, so this is how this arranges. So this is a spacer, so this is adjacent to a spacer. This purple colored thing here is adjacent to a spacer, so I'm calling this as PAM. Okay, so this PAM means protospacer adjacent motif. Okay, so this is PAM sequence. So PAM sequence is present adjacent to the spacer sequence. Okay, so what happens eventually? So now that the spacer sequence from Lord Voldemort, which is from phage, gets incorporated into the bacterial DNA. So what will happen? We all know the process, right? So DNA gets transcribed to RNA. This thing we all know. So this DNA also has to get transcribed, right? So what will happen? So there is this DNA here. So this DNA, sorry, this is a bacterial cell here, and this bacterial cell has these DNAs. Okay, so this is one CRISPR, and this is the spacer, and this is another CRISPR here, and this is another spacer here. Okay, and maybe I'll draw one more CRISPR. Okay, so this is another CRISPR here. Okay, so now I have a question for you. So this one here is a CRISPR sequence. This one here is a spacer sequence. Let me call it spacer 1. And this is another CRISPR sequence. And this is another spacer. So let me call it spacer 2. And this is another CRISPR sequence. So what is the difference between spacer 1 and spacer 2? Spacer 1 is from a different virus, and spacer 2 is from a different virus, right? So this means that the bacteria, it maintains a record of the number of viruses that has infected it previously, right? So it is like the wanted list, as I showed you before, and this is like the wanted list which this bacteria maintains. And doesn't this look like this sequence of barcodes here? So we have what is it, a sequence of black and white lines, right? So repeat lines. So doesn't this look like these barcodes here? Yeah, and that's exactly why I included those images up front. So now let's move ahead. So this spacer 2 means it means that this particular bacteria has been infected by some other phage previously, right? Okay, now let's move on to this concept here, which is DNA gets converted to RNA. So what happens to this? This is DNA, right? So this DNA has to get transcribed into RNA. So basically there will be sequences which are complementary to this sequence. Okay, and then there is one more sequence which is complementary to this sequence. There will be one more sequence which is complementary to this sequence, and one more sequence which is complementary to the sequence. I'm drawing it with the same colors. Okay, and one more which is complementary to the sequence. Okay, but now this is not a DNA anymore, this is an RNA. Okay, and now what happens is there is one more RNA. Okay, there is one more RNA. Let me draw it with some other color here, maybe this color. Okay, there is one more RNA called tracer RNA. Okay, that gets transcribed from a different locus. Okay, so this is the DNA. Okay, sorry. So this is the DNA, and from this DNA, from one another locus of the DNA, one more what to say RNA called tracer RNA gets transcribed. Okay, and I'm going to use orange color to depict the tracer RNA, and this tracer RNA is complementary to this what to say transcribed CRISPR sequence. Okay, in the RNA. So this tracer RNA This is, what is it? This is CRISPR RNA, and this is spacer 1. This is again CRISPR RNA, and this is spacer 2, and this is another CRISPR RNA. Okay, so this is nothing but a tracer RNA. This tracer RNA gets transcribed from a different locus. Since this is complementary to the CRISPR sequences in the RNA, this goes and forms a bond, or this goes and binds to the CRISPR sequence. Okay, so now we get an RNA-RNA hybrid, right? So this is nothing but an RNA-RNA hybrid. So when this RNA-RNA hybrid forms, there's something called RNase 3. Okay, this enzyme, it comes, and what it does is it simply cleaves. Okay, so this RNase will come and cleave this particular segment this way. Okay, so if you look at the site where this RNase 3 cleaves, you can see that this will result in a fragment that consists of these things. So you're going to name it. So what is this? This is spacer 1, and what is this? This is our CRISPR RNA, and what is this? This is our tracer RNA. Okay, so let me draw that. This is our spacer 1, this is our CRISPR RNA, and this is our tracer RNA. Okay, so this particular complex is formed, and this complex is very soon joined by something called Cas9. Okay, so this Cas9 is an endonuclease. Okay, so this is an enzyme which can cut or cleave the DNAs. Okay, so this structure is formed, and this structure that is it is present within this bacteria now. Okay, so let me draw the bacteria it again. So this is a bacteria. Let me draw a big bacteria, and it has its own spacers, and what is it? These repeat sequences are there. Okay, so this stretch is still there. Okay, and then yeah, it was this, sorry, we used yellow, and then it was another. Okay, so this was the DNA that was present. Let me draw. This is the DNA, and now what special structure is present? This particular sequence is present. So let me draw this now. So now there is this structure that is also present here. Okay, this one, and this one, and it is joined by a Cas9. Okay, so what is this? This is spacer 1, this is spacer 1, this is CRISPR, and this is tracer. Okay, this is present. Now let's see what will happen. I told you this is CRISPR Cas9 is a mechanism that the bacteria, what is it, performs to develop what is called immunity. Okay, so immunity means, let's see what happens if the same bacteriophage comes and infects the bacteria yet again. Okay, the same bacteriophage now comes. Okay, same Voldemort now comes. Okay, so he is now trying to infect this bacteria Harry yet again. Okay, so first time when he tried to kill Harry, what did he do? He inserted a part of himself into Harry. That's what Harry Potter is all about, right? So Harry himself became a Horcrux, right? So first, when he tried to kill Harry, Harry became a Horcrux. So a part of Voldemort was in Harry, and now what happens? Voldemort is trying to kill Harry now. So what happens now again? This part of bacteriophage DNA enters into the bacteria. So how will it look like? It will also have something called, when I said you earlier, it will have something called PAM sequence, followed by, the PAM sequence is followed by this sequence called spacer, right? So let me draw. So this is nothing but the PAM sequence, and this is nothing but the sequence which was introduced already. Okay, so now this Cas9, this is nothing but RNA, this is DNA. Okay, but this is RNA. So this RNA spacer 1, this is complementary to the spacer DNA of that of this thing, right? Bacteriophage DNA. So this will go and bind to this particular PAM and spacer. Okay, so let me draw that again. So there is PAM, okay, and then there is the spacer, and then we have the same thing, the spacer of Cas9, and then we have, okay, and then we have the CRISPR of Cas9, and then we have the tracer of Cas9 of CRISPR Cas9, and then finally we have Cas9. Okay, so this is the PAM of phage DNA. Okay, and this is the spacer or the target DNA of phage, whereas this is nothing but the spacer 1 of RNA that the bacteria has produced now. Okay, and this is nothing but the CRISPR, and this is nothing but tracer, and this is nothing but the Cas9. Okay, so now that this structure is formed, now that this complex is formed, this Cas9 being an endonuclease, it can cleave or it can cut this bacterial DNA. Okay, it can cut and inactivate the bacterial DNA. So this Cas9 introduces a double stranded break. Okay, it induces a double stranded break. So before that, let's discuss about the structure of Cas9. Okay, so Cas9 has so this Cas9 has three important domains that you have to know. So first domain is called as PI. PI means PAM interacting domain. So when I say it has a PAM interacting domain, it means that Cas9, this CRISPR Cas9 array can attack the bacteriophage only when this PAM sequence is present in it. Okay, so this PAM sequence is very much essential for this CRISPR Cas9 array to go and bind to it. Okay, and then we have this endonuclease domain. Okay, we have endonuclease domain, and apart from this, we also have one more thing called rec rec domain. Okay, so this rec domain is otherwise called as recognition domain. Okay, this is nothing but recognition domain. So this recognition domain also, it also binds to the PAM and the adjacent sequences. It holds the CRISPR Cas9 array and the bacteriophage DNA together. Okay, so this is the function of rec domain of Cas9. Endonuclease domain has two further subunits. One is called HNH and the other one is called RUVC. So this HNH part of Cas9 is the one which cleaves. So I told you the DNA will be double stranded, right? So supposing this is the target DNA. Okay, so this particular portion is the one which is targeted by Cas9. So HNH will cleave this target DNA, and I told you it is going to cause a double stranded break. So it's not enough if Cas9 breaks only one strand, it has to break the other strand also. So the other strand, the non-target strand, is broken by what is called RUVC. Okay, so this is about the structure of Cas9. So this is again the image showing the same. So it has a recognition domain 1 and recognition domain 2. I told its function, and we have the endonucleases HNH and RUVC. So this one is for the target DNA, and this one is for the non-target DNA. Okay, non-target DNA strand. Okay, and this one is called the PAM interacting domain. Okay, so this is about the Cas9, the parts of Cas9, and now and now we can see that Harry is happily going and killing Voldemort with the part of Horcrux that Voldemort himself introduced in Harry, right? The same thing. So to summarize, basically, initially I told you about the story of how this CRISPR Cas9 technology evolved. So basically, this is a mechanism of how bacteria develops immunity to these phages. So once a phage infects a bacteria, this phage will introduce its DNA sequences into the bacteria. Okay, this is a phage. So phage introduces its DNA into the bacteria. So this bacteria will have CRISPR sequences followed by certain spacers, and this spacer is how to say, this is unique to the it's a unique identifier of the virus that infected the phage. Okay, so basically this CRISPR spacer sequence gets transcribed and you get RNA, and to this sequence of RNA there is one more RNA that gets attached, that's called as tracer RNA. So this RNA hybrid is cleaved by RNase 3. So this RNase 3 digestion gives you a complex that is made up of a spacer, a CRISPR, and a tracer, which is very soon joined by Cas9. Cas9 is an endonuclease. This Cas9 has three major domains. One is PAM interacting domain. I've shown you here, right? The PAM interacting domain, the endonuclease domain, and the recognition domain. And once this Cas9, this particular array is formed, this keeps what to say, this is maintained within the bacterial cell. So once the bacteriophage, again, if the bacteriophage is going to reinfect this particular bacteria again, this new DNA sequence of the phage is cleaved by this CRISPR Cas9 array. Okay, so this is about how CRISPR Cas9 helps a bacteria as a tool of immunity, right? So now let's see how this is exploited as a gene editing tool. This was actually further developed by these two ladies here. I hope you know these two. This is Emmanuel Charpentier and Jennifer Doudna, right? So this Charpentier and Diona. So these two ladies, so they found that, or they exploited this concept, and they kind of used it in clinical scenarios to find a cure to all the elements, especially point mutations, diseases caused by point mutations. So one example is sickle cell. Okay, so let's see how CRISPR mediated gene editing can be used as a gene editing tool for especially sickle cell anemia. So before that, see, CRISPR, let's talk about sickle cell anemia. So in sickle cell anemia, what happens is there is HBS. Okay, so this HBS has the oxygen carrying capacity is like very less, and so the as a therapeutic intervention we try to increase HBF, right? So HBF actually basically what happens from birth, okay, from the late fetal life, okay, from the late fetal life till birth, which hemoglobin fraction is overproduced, it is always HbF, right? But after birth, what happens is following birth, HbA predominates, okay, HbF is suppressed, right? But because in HbS is actually, instead of HbA, HbS is produced more. So the oxygen carrying capacity is very less. So in order to enhance the same, HbF is stimulated. Okay, we try to stimulate HbF production, right? But what happens is in sickle cell, after birth, not in sickle cell, in any person for that matter, after birth, there is one particular transcription factor called BCL11A. Okay, you can see that there is BCL11A here. Okay, this BCL11A is a transcription factor. So this BCL11A after birth, okay, after birth, BCL11A transcription factor is increased. Okay, so this BCL11A will decrease this HbF production. Okay, this suppresses the expression of HbF. So what they're trying to do is they're trying to target this BCL11A. So once this BCL11A is destroyed, if you destroy this BCL11A, this inhibition over HbF is removed, and now HbF can be expressed enormously, right? So this was their idea. So what they did is they developed. So supposing there is supposing this is a BCL11A gene. Okay, so this particular in this particular case, they tried to develop a guide RNA. Okay, this is called a guide RNA, and this is complex to Cas9. Okay, and this guide RNA, it has sequences which are complementary to the target nucleotide. Okay, complementary to the target nucleotide. In this case, it is BCL11A. So it is, this guide RNA is complementary to this BCL11A sequence. So this goes and binds here, okay. And once it binds here, what is the function of Cas9? Cas9 is an endonuclease. It brings about double-stranded break. Okay, so let me draw one more strand here. So this brings about double-stranded break. Cas9 can bring about double-stranded break. So once there's a double-stranded break, what happens? So once there's a double-stranded break, what is a repair mechanism that our body tries to adapt? So there are two, for double-stranded breaks, there are two repair mechanisms that are possible. One is non-homologous end-joining pathway, and the other one is homologous recombination. But in this given scenario, the body prefers to go in for non-homologous end-joining pathway because there is no complementary or no template DNA that is available for repair. So what happens? Basically, there are two strands here, okay. There are two strands here and there is a double stranded break, right? So since there is no complementary strand, basically the body doesn't know which sequences, whether to incorporate A followed by G or then followed by C. So it doesn't know the sequence, the order, and the nucleotides, number of nucleotides that has to be put in to fill this gap, right? So it randomly keeps adding these nucleotides, and this random addition of nucleotides can result in either insertion or deletion kind of mutations, which in turn can give rise to frame shift mutations. So this, as a result of these so many mutations put together, this particular gene will be, what to say, faulty, okay. So even if there is a protein that is produced as a result of this faulty or this faulty or mutated gene, now that's going to get degraded, okay. So the protein that is produced as a result of these mutations again is going to be faulty, and hence it's going to get degraded. So this is how they wanted to, suppress or inhibit BCL11A. Okay, so once this BCL11A was inhibited altogether, HbF production increased. Okay, so there is one more way how you can address the sickle cell here. That is, here we all know that sickle cell is due to a point mutation. So this point mutation is at sixth position of beta globin gene, and in beta globin gene, in sixth position of beta globin gene, what goes and what comes? Glutamine goes, okay, glutamine goes, and instead valine gets incorporated, right? So I'm not sure what the codons are. Let me just give you a scenario. Supposing GAG is the codon for glutamine and GTG is the codon for valine. What happens? There is this point mutation GA. So this A is replaced by T, and that is why there are two different what to say amino acids that mediate the disease, right? So what we can do is that, along with this CRISPR and Cas guide RNA, okay. So here I told you guide RNA, but whereas when I discussed to you about the bacterial thing, there was a sequence of RNAs. There was a spacer, and then CRISPR, and then tracer. So replacing all these three together, we have something called guide RNA here in CRISPR Cas9 technology. Okay, so here in this scenario, we can not only, like, we have this guide RNA, okay, we have this guide RNA and Cas9, right? So in addition, maybe we can also supply a template sequence. Okay, we can also supply a template nucleotide sequence. Okay, template nucleotide sequence. So this template nucleotide sequence will have sequences which are complementary to the sequence that has to get incorporated. There is this, imagine this is the two strands of 3' to 5', 5' to 3' of sickle cell gene, okay, sickle cell gene, and there is this point mutation here, so addressing which we developed a CRISPR Cas9 technology based guide RNA. This guide RNA identified this thing, and this Cas9 now brought about double stranded break. Okay, so now there is a double stranded break, and okay, 3' 5' 5' and 3 dash. Okay, so now there's a double stranded break, but along with this Cas9 we have also supplied a template DNA. Okay, we've also supplied a template DNA, which means the body, now the body goes in for this, utilizes this is a template DNA, 3 dash and 5 dash, this is 5 dash and 3 dash. So this is a template DNA with the sequences which you want to incorporate into. So now the body relies on homologous recombination more than non-homologous recombination. Okay, non-homologous recombination is used only when there is no template DNA that is present, but in this case maybe we can give a template DNA. So now the body goes in for homologous recombination. So what happens in homologous recombination? There's something called strand invasion. So this strand goes and invades, right? So let me draw. This strand goes and invades. Okay, so like this, right? Okay, so this strand will act as a template so that new nucleotides are synthesized here, right? This is how this homologous recombination works. And as a result of homologous recombination, we can bring in, or we can edit the DNA, the mutated segment of DNA. So instead of instead of GTG is the mutated sequence, we can bring in GAG. Okay, so instead of valine we will get glutamine. Okay, so this is how CRISPR-Cas9 technology works. This is all about CRISPR-Cas9. So initially we started with the origin, the story, the interesting story of how CRISPR-Cas9 technology evolved. So it started with Ishino, and then Moshiko, and then this Philip. So this Philip story was interesting. This Danish yogurt story, and then how this bacteriophages, while infecting the bacteria, insert a part of their DNA sequences into the bacteria, and this how this bacteria employs the same as a tool or a defense mechanism against the bacteriophages when it is subjected to a secondary attack by the same phage. So that we saw. And basically this what to say, CRISPR-Cas9 complex is made up of, in case of bacteria, it is made up of a spacer and a CRISPR and a tracer DNA, and then we have this Cas9. Okay, so this is it. And one more important thing, Cas9 and its subunits. So we have recognition subunits, we have PAM subunits, which is protospacer adjacent motif interacting subunit, and then the endonuclease subunit. And this PAM sequence is very much important. Only if PAM sequences are present in bacteriophages, these CRISPR-Cas9 can go and bind to it. Okay, if not, they cannot bind. Fine. And now this is all about how CRISPR-Cas9 works in case of a bacteria, and now this is exploited as a gene editing tool as well. So with regard to gene editing tool, I give you an example of sickle cell, how it works in sickle cell anemia, how CRISPR-Cas9 can be used to silence or to inactivate a particular genome. So there we go in, the body goes in for non-homologous end joining mechanism, and if you provide a template strand, the body goes in for homologous recombination and then corrects the mutation as well. So this is all about this interesting concept of CRISPR-Cas9 technology. I hope you all enjoy this particular topic. This is personally my favorite topic too. So keep revising. All the best.